Bismillah ar-Rahman ar-Rahim 9700. This is the November 23 series which we are going to start today. It's paper 1-1 is an MCQ and we'll probably finish this in 2 videos. Please remember to pray to Allah every day. O Lord, give us good in this world and good in the hereafter and defend us from the torment of the fire. Now, as we read question number 1, until recently the typical viruses known to science were 20 to 150 nanometer in size. In 2003, the Mimi virus was discovered with a size of approximately 680 nanometer. In 2013, the Pandora virus was discovered, which has a size of over 1000 nanometer. Which viruses can be seen using a light microscope with a maximum resolution of 2.25 micrometer? Now, basically, they want you to convert this into nanometer. Now, you know that 1 micrometer is equal to 1000 nanometer. So, if you multiply 0.25 into 1000, well, you get 250. So this resolution is 250. That means you cannot not see anything less than 250. Now you know this one is only less than 250. The rest of it are all are more than 250. Now it says which viruses can be seen using a light microscope with a maximum resolution of 0.25. So that is why the answer is B. You cannot see this virus, but you can see these and these. Because the others, it said both. It didn't say just light microscope. So it says using a light with a maximum, and the and is very is written in a way that you have to insist on. Everybody wants you to look at that. So the question number 1 is B. It says viruses can be seen using a light microscope. If it said only a light microscope, then only this one you would have been able to see. Of course, the bigger ones you would always see, but it says you can see with using a light microscope with a maximum resolution of 0.25. Electron microscope, of course, can see anything because it has a very high resolution. It has a very high resolution. Now, coming to question number 2, I know this is a little difficult. Some of you find this very difficult. I wish I could individually explain this to you. Some of you, of course, can figure it out. Let me try my best to explain it to you. The diagram shows an eyepiece graticule and cell viewed through a microscope. When the eyepiece graticule was calibrated, so this is basically an eyepiece graticule. We call it EPG, eyepiece graticule. When the eyepiece graticule was calibrated at this magnification, the whole length of the graticule shown covered 10 divisions. So whole length of it, 100 EPG, is equal to 12 stage micrometer. 12 divisions of the stage. But then they also tell you that there were 100 divisions in the stage, and that was 10 millimeter of the stage micrometer. So 100 divisions is equal to 10 millimeter. First, I'm going to do that calculator. So 1 division is equal to 10 over 100 is equal to 0.1 millimeter. So I first done this working. And then it said, okay, so 100 EPG is equal to 12 into 0.1. That is equal to 1.2. Then how much will 30? Because here you can see this cell is about 30. So 30 will be equal to 0.366 millimeter. But there is no such answer here. There's only one answer, 3 millimeter. The rest of them are all in micrometer. So you multiply it by 1000 and the answer is C. First you have to do the calculation. 100 is equal to 10 millimeter. This is the first calculation that I did. So 1 is equal to this. So 100 EPG were equal to 12. So 12 into 0.1. So that's equal to 1.2. And then 30 would be equal to. So 30 of these would be equal to this. And this is how you do the working. Please do this paper before you actually see my video so that you can put your mind to it as well and see where you're going wrong. Then coming to question number 3, in Tay-Sachs disease results in a buildup of lipids in cells. Which cell structure does not function correctly in this disease? For lipid synthesis, what do you need? You need mitochondria. For anything synthesis, you need mitochondria. You need smooth endoplasmic reticulum. You need the Golgi body to process it, to modify it. So what do you not need? You do not need the lysosome. Lysosomes are hydrolytic enzymes. We don't need them. The buildup of lipid means the lipid is not being processed. So that is only done in the smooth endoplasmic reticulum. And in order to do that, just like for protein synthesis, you need mitochondria, you need ATP, you need energy. So mitochondria is needed for everything. Protein synthesis, lipid synthesis, from glucose to glycogen when it's converted in the liver, even that you need the ATP. Question number 4. Which animal cells would have the most extensive Golgi body? Golgi body does what? Adds a glycogen part. Adds a glycogen part. So where will glycoproteins be made? Glycoproteins. Which glycoproteins have we studied? Mucus is a glycoprotein. Antibodies are glycoproteins. So antibodies are produced by lymphocytes. Another question I could give you on that. So basically what you have to understand is the goblet cells which will produce mucus, and mucus means it's a glycoprotein. So the proteins would be made on the ER, on the rough endoplasmic reticulum. There'll be more ribosomes because mucus, more ribosomes. And then they have glycogen part added by the Golgi. So it says which animal cell would have the most extensive Golgi body. I give you a simple example. In your house, you have one stove or some sort of electric or gas stove to cook the food. When in a restaurant, there'll be 20 or 30 of them because a lot of food is being cooked. Then coming to question number 5. The diagram shows 3 circles 1, 3 and 5 and the shared structures 2 and 4. Which row correctly identifies 3 circles and some of the structures that are shared between them? Have a look at it, think about it. Now basically you've got to understand they're comparing what? Chloroplast, prokaryotes, mitochondria. Now prokaryotes have circular DNA, they have 70S ribosome. Mitochondria also have circular DNA, 70S ribosome. Chloroplast also have circular DNA and 70S ribosome. So basically the answer is C. Why is the answer C? Prokaryotes have circular DNA, mitochondria also have circular DNA, they share it between 1 and 3. And mitochondria and chloroplast also share 4, which is circular DNA. What was wrong here? ATS was totally wrong. Because chloroplast, mitochondria, prokaryotes do not have ATS ribosome. So I would have cancelled these out immediately. These were the wrong ones. And I was only left with C and D. But then it said here again it's ATS. So that was a very easy way of cancelling it out because they were comparing only chloroplast, mitochondria and prokaryote. And all 3 of them do not have any ATS ribosome. So the ATS, we should have cancelled them out in the beginning and said, okay, that's all rubbish. Now coming to question number 6, which row about the genetic material in animal cells and prokaryote cells is correct? Now this is first chapter. Either you know this or you don't know this because you've not been memorizing it. You have to learn a lot of things. You have to memorize. आपको बहुत चीजों का रट्टा लगाना है. You need to ratify a lot of stuff. Animal cells contain linear DNA. Yes. So D is totally wrong. Prokaryotic genetic remains surrounded by a double membrane. That's incorrect. So only these two can be correct. Prokaryotic genetic material is double-stranded DNA. Yes, it is double-stranded DNA. So this can be correct, this can be correct, and this can be correct. So which row about the genetic material? So it becomes very easy if you know it. So the answer is C. Animal cells always contain linear DNA in a way that's in I can put another MCQs contains linear DNA. It didn't say always because there is circular DNA in the mitochondria of an animal cell. Then prokaryotes doesn't have a nuclear membrane. There is no nuclear membrane. That's why it's called prokaryote, no nucleus. And prokaryotic genetic material is double-stranded DNA. Please remember, all DNA that we studied is double-stranded. Whether it is circular or whether it is linear, it is all double-stranded DNA. You will not study it till A2. You will study in that viruses have single-stranded DNA, but that's just I'm telling you that information, not relevant to you at the AS level. So this is what you have to understand is that animal cells contain linear DNA. Yes, in the nucleus they contain linear DNA, but in the mitochondria they contain circular DNA. Now, what is present in all viruses? Now, why are they asking you these questions? In your AS syllabus, viruses are 2 types, DNA and RNA. So what would be? Uracil is only present in RNA. It says all viruses. Ribose is only present in RNA. You see, DNA has deoxyribose and it has bases ATCG. RNA has ribose and it has AUCG. So what is common to both is the G part. Guanine. Right? DNA has deoxyribose, RNA has ribose. So ribose was correct. Thymine was wrong because thymine is only present in DNA. Viruses are of two types, either containing DNA or containing RNA, and also they have a protein coat in all of them. And some of them have the phospholipid layer outside it. Some of them have the phospholipid bilayer outside it. So this is how you had to understand this part of the question. Now coming to question number 8, the test for non-reducing sugar requires a second Benedict's test to be carried out. Which set of steps is the correct method for carrying out the non-reducing sugar test before carrying out the second Benedict's test? Non-reducing sugar requires a second Benedict's test. So we have to first do the Benedict's test, and then it's going to be negative. It contains sucrose. And then we do the second test in which we add HCl, heat, cool, add alkali, and then add Benedict's. So the first test, the Benedict's test, has to remain blue. So which set of steps is for carrying out the non-reducing test before carrying out the second Benedict's test? Those tests that we use, first we've done that test and it's negative. So what do we have to do before we add the Benedict solution? We have to perform the Benedict's test with the color of the solution remaining blue, boil with dilute HCl, neutralize with sodium hydrogen carbonate. Perform the Benedict's test and it is blue. That shows there is no reducing sugar because there could be glucose and sucrose. Then you boil with dilute HCl and then you neutralize with sodium hydrogen carbonate. So warm was wrong here. Now this, of course, if it's showing red, that means the Benedict's was present. That means some sort of reducing sugar was already present. That's why this was wrong. Perform the Benedict's test which gives a negative result. What does a negative result mean? And then it says neutralize with sodium. This is all the steps have gone wrong. It says neutralize with hydrogen carbonate and gently with HCl. So HCl comes first. Neutralize with HCl with alkali comes after that. Question number 9. The diagram shows three types of covalent bonds. Which bonds will be found in glycolipids and glycoproteins? Now here is the glycoprotein. This would be a bond in the glycoprotein. So you have to understand is bond 1, bond 2 and bond 3. Now the answer is A because glycolipids only. This bond will only be present in glycolipids. The bond 2 will be found in both glycolipids and glycoproteins. And bond 3 would be found in glycoproteins. Now coming to question number 10. Which molecules will be found in an oil, which is a liquid, and more than in a fat? Now if you look at a fat, solid, that means it's a saturated fat. And oil liquid means it's an unsaturated fat. So I did all the different types of bonds here. So I said this 18, 18, 18, these ones, I added these. And then I added these. And then I saw, because you see, this will tell me how many of them are saturated and how many of them are unsaturated. And then here this was 14, 7, and 4. So these had less double bonds. You see, saturated fats have single bonds and unsaturated fats have double bonds. Saturated fats have single bonds and unsaturated fats have double bonds. So 2 and 4 were the least ones, and that is how I gave it. That's how I figured it out. I know my chemistry is not very good. Maybe your chemistry is better than mine. I mean your understanding of chemistry. I don't mean your chemistry. Question number 11. Which row about the bonding found in the primary, secondary, tertiary, and quaternary structure of protein is correct? Now, primary structures are all peptide bonds, but peptide bonds are also covalent bonds. You have to know them. There's a special point in the syllabus, the type of covalent bonds. So, all these was correct. Primary was all correct, but secondary here we find there's the hydrogen bonds in the secondary structure, which forms the alpha helix and the beta pleated sheets. And then in the tertiary structure, we have the hydrogen ionic, covalent, and hydrophobic interactions. And quaternary also we have the same of them. So that is why the answer is B. Hydrogen ionic, covalent, and hydrophobic interactions and disulfide bonds as well. So they are the four bonds. Hydrogen ionic, covalent, they've said to call it the disulfide, covalent, and hydrophobic interactions. Question number 12. The specific heat capacity of different substances is shown. Air is 1.0 specific heat joule grams per degree Celsius, hydrogen 14.3, water 4.2, and water steam 2.1. Which statement is correct? Air is more stable environment than water because it is more resistant to changes in temperature. It takes more energy to raise the temperature of hydrogen gas than it does to raise the temperature of water. A specific heat capacity of 4.2 joules means that it takes 4.2 joules of energy to vaporize 1 gram of liquid water. There are more hydrogen bonds between water molecules in a gas than between water molecules in a liquid. You see, the water becomes a vapor. There are no hydrogen bonds. So it was easy to say which ones were wrong. So that is why then you could figure out that B was the only correct answer, which made sense. Others were all wrong somehow or the other. Then coming to question number 13, the solid line on the graph. Which one is the solid line? Let's look at the solid line. The solid line on the graph, this is the solid line. It goes up to here. The solid line on the graph represents the product formed over time for a reaction in a cell. Amylase, starch, maltose being formed. So total product formed, and this is time. So initially then we working on starch, and it is being converted by amylase into maltose. So the maltose is less in the beginning, and slowly it increases. Which other line represents the effect of adding the enzyme for this reaction? For adding an enzyme for this reaction. What do enzymes do? They lower the activation energy. They lower the activation energy and the reaction will occur very fast. The total form of the product will be more. So which other line? A, B, C, or D? Now it's very easy to figure it out. It had to be A. It had to be A. It couldn't be anything else. The product would be the same because the starch is 100. The starch was 100 initially also. So the maltose which is produced says 1000. So the B and D were totally wrong. The only confusion was between A and C, but C, the rate is decreasing. So the total product formed would be at 1 minute, the total product formed with an enzyme would actually be more. But then the product at the final end should also be the same. It can't be more. B and D was wrong because the final product was more or less. B and more, D was less. So which other line represents the adding the enzyme for this reaction? So the answer is A. Question number 14. The graph shows how the rate of enzyme controlled reaction is affected by the change in temperature. All other variables were standardized. Rate of reaction, region X, temperature. We have temperature on the y-axis, rate of reaction here. I always say whatever is on the x-axis, whether it is photosynthesis or limiting factor, it's always what is on the x-axis. What is the factor limiting the rate in region X? So the answer was very simple. It was C. Because temperature is limiting the factor. Temperature was 10 here, temperature is 15 here, 20 here, and the rate is increasing. Up to this point, the optimum is reached. So before the optimum, the temperature is the limiting factor. Before the optimum, the temperature is the limiting factor. Then after the optimum, of course, the enzyme denatures. So the active number of active sites would be the limiting factor. Now, question number 15. How will the removal of a reversible non-competitive enzyme inhibitor affect an enzyme controlled reaction? Now, the Km will decrease. Now, first of all, let's understand Km. Now, I'm going to go back and then I'm going to come back to this MCQ. Now, if you look at here, effect of substrate concentration on the rate of reaction. What is Vmax? Vmax is the maximum rate of reaction at the point where all the active sites are bound to substrate, enzyme is saturated. Michaelis-Menten constant Km is the substrate concentration. It is the substrate concentration at which it is equal to half of the maximum value, that is half Vmax. So we have calculated the Vmax. This is the Vmax, whatever it is, say it's 100 here. So half of that would be 50 here. And then you draw a line and then you see what is the Km. So please first understand this. What is Km? Km is the substrate concentration which is equal to half of its maximum value. If you look at now, if you look at these, in a competitive inhibitor, the Vmax remains the same. In a non-competitive inhibitor, the Vmax decreases. But they've said remove the non-competitive inhibitor. So if you remove the non-competitive inhibitor, the Vmax will increase. When you remove the non-competitive inhibitor, the Vmax will increase. But what will happen to the Km? Let's look at that. You can see from this graph. You see, when you've removed this non-competitive inhibitor, you've removed the non-competitive inhibitor, you remove this one. So the Vmax will increase, but the Km stays the same. You see, here, this was the Vmax for the non-competitive inhibitor. This is the half Vmax. So the Km is this. And for this, when this was the Vmax, this was the half Vmax. It's the same color. You can see it's written in black marker and red marker. So the Km remains the same. Please understand this. I don't want you to just memorize it and say, oh, well, this is the answer. No, for me, every student must understand every question. I take all this effort to make these videos to make you understand it. Now let's read the question. How will the removal of a reversible non-competitive inhibitor affect an enzyme catalyzed reaction? So the Vmax will increase. There were only two possible answers. Vmax will increase. So the answer is C. Why? Because the Km will not change and the Vmax will increase. It said reversible, removal of a reversible. It didn't say the other way around. Question number 16. The dimensions of three agar cylinders X, Y, and Z are summarized in the table. Cylinder X is 2 millimeter radius and length is 10. So 2 millimeter is the radius and the length is 10. Then it's 1 millimeter and the length is 20. And then it's 5 into 5. It's a cube. Now it says, what is the correct order of surface area to volume ratio for the cylinders from the smallest to the largest? Now I, of course, did it in my own little way, but the answer is C. Of course, you can do it separately for all of them and calculate the surface area to volume ratio. We know how to do all that calculation. We've done it in a number of paper threes. And you can easily do that and then calculate the surface area to volume ratio. You must know how to do that. So question number 16, the answer is C. Question number 17. Three individual plant leaf cells were each placed in a different solution for 30 minutes. Each solution had a different water potential. Which row correctly shows the change in volume of the three cells? You see, when the water potential of the solution was lower than the cells. What is water potential? Free water molecules. It said water potential was lower than the cells. So what would happen? Water would move out and the volume of the cell would decrease. Now, why is the answer B? Because it has decreased when it is equal to the cell. If it's outside also 2%, then there's going to be no net movement of water. 10 molecules of water moving in, 10 moving out. So there's going to be no change. So the only no change were correct. And if the water potential is higher, that means it's a dilute solution outside. The cell is 2%. This is the cell which I've drawn for you. If the water potential is higher, that means it's a dilute solution, more free water molecules. So water is going to move in and this is going to increase. Question number 18. Which row is a representation of the chromosomes at the beginning of prophase of mitosis and the number of DNA strands in the chromosome? You see, this is one chromosome, one DNA molecule, but it has two strands of DNA, ATGC. Now, the appearance of the chromosome in prophase, interphase has taken place. S phase of interphase, the DNA is replicated, so it can only be this and this. And the number of strands, this has got 2 strands. Now, when another copy of it is formed, it has 4 strands. These are 2 strands in this, 2 strands in this. This is this one thingy. This is this one thingy. So that is why the answer to question 18 is B. Appearance of one chromosome and number of DNA strands is 4. Then very basic, which events are part of mitosis? You know interphase is not part of mitosis. You know cytokinase is not part of mitosis. So the answer is only telophase. Mitosis is only prophase, metaphase, anaphase, and telophase. The rest of it is all part of cytokinase is separate. Interphase G1, S, and G2 is part of interphase. Question number 20. The statements are about genes and proteins involved in breast cancer. So a person who will have, the question is, which combination of genes is most likely to result in breast cancer? The protein coded by the BRCA1 gene inhibits the growth of breast. So if you have that, it is going to inhibit. If you have the protein coded by the if you have the BRCA1 gene, well, you won't have cancer. The protein coded by the P53 gene suppresses tumors. Which combination of genes is most likely to result in breast cancer? Well, if you don't have either of them, well, then of course you're more prone to breast cancer. This is one of the genetic testing that we do now. Then it says question number 21, the photograph shows a cell during mitosis. What is happening in the cell? Chromosomes are condensing, centromeres are moving to the opposite poles, spindle microtubules are shortening. It's not asking you the stage, it's saying what is happening. So the answer is C, 2 and 3. Chromosomes are not condensing, they're just moving to the poles. Centromeres are moving to the opposite poles and spindle microtubules are shortening. Chromosomes are condensing in, they start in telophase and then will finish off in cytokinesis. Will condense, they will condense in prophase. They will go back to the thin thread-like structures when they go back into telophase, and then of course the two cells are formed. So they're not condensing, they're condensing only in prophase, shorten and thicken. Which diagram correctly shows the structure of a piece of DNA? So what can you not have in the DNA? You cannot have U. So let's remove all the U's first. If there are any more U's, no, there aren't any U's. So it has to be ATGC, so it can't be TG. That's wrong. What else is wrong? ATGC. It can only be ATGC. It can't be anything else. Why is this wrong? You see, these were in these direction. They're anti-parallel. These have to be in this direction. These were in this direction. These have to be in this direction. They are both in the same direction. So this was wrong. They're anti-parallel. The deoxyribose is pointing in the other direction. So that is why the only one correct is D, and the answer to question number 22 is D. You can see the D is correct. This is in this direction, this is in this direction, and ATGC and all is correct. In this video, we have discussed question 1 to question 22 and we'll continue in the next video.